welcome to biology class. We have a special guest today. This is Max, who likes to hang out in jackets. And we are going to talk about pedigrees, and we're going to use the example of achondroplasia. And if Max makes an appearance, we'll just let him make an appearance. All right, so I'm going to share my screen. Achondroplasia is the most common form of dwarfism in humans. It affects a gene that controls long bone growth. Long bones are the bones that we have in our arms and our legs. Typically, people with achondroplasia have average-sized torsos, but shorter arms and legs. And it is the most common form of dwarfism. So about 90% of dwarfism is achondroplasia. Achondroplasia is interesting in our class because we're talking about genetics. It is a genetic disorder. It's linked to a dominant allele, and it's autosomal. Autosomal means it's one of the chromosomes 1 through 22. Those are autosomal chromosomes. The 23rd chromosome, X and Y, are sex chromosomes. And we'll look at examples of those in a second. So here we have a visual. This is our friend Jason Acuna, the actor and stunt performer. And this photo was taken in Portland, Oregon in 2009. And it's from WikiCommons, which is usable without copyright infringement. So let's look at how achondroplasia as an autosomal dominant trait is inherited. If you have an unaffected parent, they do not have the dominant allele. So they must have 2 recessive alleles, 2 because humans are diploid. If you have an affected parent, they're going to have 1 dominant allele and 1 recessive allele. Now, with achondroplasia, if you get 2 dominant, if you are heterozygous dominant, the embryo does not develop. And if it does turn into a fetus, that child unfortunately dies shortly after birth. So in this disorder, there is no heterozygous dominant that is viable. So we have homozygous, recessive, or heterozygous. If these 2 parents were to have 4 children, they would statistically, based on a pedigree, have 2 unaffected children and 2 affected children. And we could do a Punnett square to figure that out pretty quickly. But what we want to do for our purposes today is work on pedigrees. So let's look at this in a pedigree. For pedigrees, we denote people as shapes. So males are squares and females are circles. And forgive the binary, we know that there's way more variation in real life, but we are simplifying and we're working with chromosomes, so we're basing this on chromosomal sex. If your shape is not filled in, then you are unaffected. If your shape is filled in, then you are affected. I'm going to see if I can have a better way to fill this in. Yeah, there we go. So it is solid. It is a filled in shape. That would be an affected person. So for our offspring, we have 1 girl with achondroplasia. So she would get a filled in circle, and 1 boy with achondroplasia. He would get a filled in square. And our other 2 children are unaffected, which means they would have a square for a boy and a circle for a girl. If we took this and moved it over and connected the parents like so, and then drew a line down and added it with the children like this, we have a pedigree. So that's the idea behind a pedigree, is you're representing the people in the condition with the circles and whether or not the circles are filled in or not. I'm going to erase my drawings so we can go to the next slide. So picture what I had just drawn, and now we can see a dominant trait in a family. So here I have a square. If it's a square, what does that mean? This is going to be a male. And here I have a circle, so that's going to be a female. This square is not filled in, So this is going to be unaffected. Anybody that is filled in is affected. So this is going to be an affected male right here. And this is going to be an affected female. We say affected because we can use this to look at many different types of traits. Maybe this is a chondroplasia. Maybe it is having brown eyes or having curly hair, whatever it is, that trait would be the one you're looking at, would be the affected one. So it doesn't always have to be a disorder, but we started using pedigrees to track disorders in family, which is where the vocab came from. Now, what's really cool about pedigrees is we can track multiple generations at once. So here's the first generation. These would be the parents of the next generation. So generation 2 is the children of the parents. And then the third generation, these are the grandchildren of the first generation. So grandparents, parents, grandchildren. It's 3 generations. We start with 2 individuals, and the line down shows their number of offspring. So they have had 2 girls and 1 boy. They had 3 kids total. Their first girl had children with this male, and they had 2 daughters. Their second child was a boy with the trait. Let's say it's a chondroplasia. This boy had children with this female, and they had 1 boy and 1 girl. Both of them have a chondroplasia. Their last child is a girl. She had children with this male. She had 2 sons, and one of those sons has a chondroplasia. These kids in relation to each other are cousins. So 2 and 3 are cousins to each other. If we look at number 2 and this male right here, he is this girl's uncle. Just to try and get you family oriented, because his sister's kid is number 2. When we look at pedigrees, we can also use them to figure out the genotypes. The phenotype is already given because of the coloring in. So the coloring in tells me this person has a chondroplasia. I know chondroplasia is caused by a dominant trait, so they have to have at least 1 big A. This person, because they are unaffected, I know their genotype is recessive, and to have a recessive genotype and phenotype, I have to have 2 little a's. So this person has little a, little a. I told you earlier that a chondroplasia is fatal if there's 2 dominant. So I know that this individual, because they lived long enough to have kids, has to be a heterozygous. So if I look at the offspring down here, I can automatically start to fill in their genotypes based on their phenotypes. Any child without a chondroplasia is going to be homozygous recessive. So all of these people without a chondroplasia must be homozygous recessive. That's going to be true in the 3rd generation too. How do I know that? Because they do not have the trait, and it's a dominant trait. Dominant traits will show up in every generation. They will affect men and women equally. Now, if this was something else and I could have homozygous dominant, I would have to be looking at who they're mating with to have a prediction of their genotypes. But for this, I can tell that every filled in circle is going to be heterozygous, because achondroplasia is a heterozygous condition. So I can fill in the genotypes of all of my individuals. Let's look at a pedigree for a recessive trait. Here's a recessive trait. I have 3 generations again. Here are the grandparents. They had 2 children, 1 woman, 1 boy, 1 male. This woman did not have any children, so there is no generation below her. She could be married, totally happy, everything like that. This is not about your relationships. It's about your passing on of genes and alleles. So here, this male passed on his genes with this female. They don't have to be married, like it's just passing on genes. And they had 3 children. So I've been using the relationship grandfather, grandmother, all that. But it's also important to think that this is just biological traits, not our impositions of how these interactions have to be. So here, this male and this female, they had 2 girls and 1 boy. And of their children, 2 of the 3 have this trait, but the 3rd does not. And neither of the parents has the trait. So whenever you see 2 parents that do not have the trait producing a child that does have the trait, you know that that's going to be a recessive condition. And why does that happen? Let's draw out the genotypes and that will help us understand. So this male is going to have at least 1 dominant allele. I'm going to write big R so that we can just make it easy to see. This female also has 1 dominant allele. Because they have that dominant allele, they are not expressing the recessive trait. They, however, have 2 children that are expressing the recessive trait. So to express the recessive trait, you can only have recessive alleles. So both of these kids must be homozygous recessive. Now, if these kids are homozygous recessive, they must have got 1 of these little R's from each of their parents. Like they can't get an allele that their parents don't have. It had to come from their parents. So I now know that each of these parents is heterozygous. Because these parents are heterozygous, they both passed on their little R, so their offspring were born with a condition that they themselves do not have. Now, their daughter over here, she has at least 1 big R because she does not have the recessive condition, but she could be a heterozygote, or because both her parents have big R's, she could be homozygous dominant. We don't know. And we won't know unless this person decides to have children. And if they have children, let's say they have 3 of them. The first child is a girl, no condition. I still don't know her genotype. Second child is a girl, no disorder. I still don't know her genotype. But if at any point this daughter has a child that does have the recessive condition, so I'm going to fill that in. Now I know that the mom has to be heterozygous because that little R had to come from somewhere. So that little R got passed down to that offspring. If this daughter here never has children, then we will never know whether or not this child is homozygous or heterozygous. Erase those. So in dominant pedigrees, you have the generations, every single one of them showing the trait. That's your indicator that it's a dominant allele and it's affecting men and women equally. In recessive traits, you have parents without the condition producing children that have it, but it affects men and women equally. And in our 3rd pedigree, sex-linked traits, now we have our parents that are unaffected having children that are affected. But look, it's only men. So this is an disequal association of the trait with one sex over the other. And the reason is because it's usually on the Y or X chromosome. It's almost always on the X chromosome because the Y chromosome itself is small and it doesn't have that many genes. It has really important genes, the main one being the SRY gene that codes for maleness, which means that if your embryo has it, they often develop a penis and testes and testosterone at higher levels than if you did not have that gene. But that's not the only thing that determines sex, as we know, it's highly variable. So in this trait, we're going to link whatever it is that we're looking at to a X chromosome, because it's bigger, it's more likely it's going to be on the X chromosome. Males have an XY genotype. Females have an XX genotype. But because the Y is usually not carrying anything, it's on the X chromosome where we're going to write the allele. So something that often affects men more than women is color blindness. So we're going to make color blindness our trait. If you are not colorblind, you have the dominant version. So here's a not colorblind male and a not colorblind female. But the female also has the recessive allele. We refer to her as a carrier. She can pass this on to her kids, but she herself is not colorblind. So let's look at what happens when these two people have 3 children. They're going to produce 1 boy and 2 girls. Now, both of these girls are not colorblind, so they have the X with the big B, big B, that has determined them being not colorblind. This male is colorblind. Because this person is a male, they inherited the Y chromosome from their father. That Y chromosome did not have an allele for color blindness on it. So the chromosomal sex of your baby is determined by the sperm. The reason it's determined by the sperm, by the father, is because the mother does not have a Y to pass on. The father can put either this chromosome or this chromosome in his sperm. And so to make a boy, he had a Y chromosome in that sperm, which means that this colorblind allele came from the mother. Now, this individual here gets a second X allele, and that X allele could be big B or little b. We're not sure until we know later on whether or not this person has a colorblind child. Here, we can determine the genotype of this person based on their offspring. So individual in generation 2, individual 5, produced a child that is colorblind and is a male. That Y had to come from dad. So here's dad. But dad himself is not colorblind. So his X chromosome contains the big B. That means for this child to be colorblind, the recessive allele had to be carried by mom. Because if it was from dad, dad would be colorblind. That means that this child here, were he to have a kid, the X chromosome he passes on will have the colorblindness allele. So any of his daughters have a chance of getting colorblindness from him. His sons will not, because he only has the Y allele to pass on the sons. So it's really the mother's recessive allele that causes the colorblindness. The reason it affects men more than women is because women have 2 chances to hide it, right? So if I inherited just 1 X from my mom, it's a 50-50% chance that I would get this colorblindness gene. But since I have 2 X's, I get one from my dad as well. Now I have a chance to hide my recessive allele and hence the carrier. Cool. So when you're looking at pedigrees, if it's skipping generations and showing up from parents that do not have the trait, but it's only men that are getting affected, it's a sex-linked recessive pedigree. Let's look at a practice one. Based on this pedigree, what type of inheritance is shown? What is the genotype of individual 1, 1, and what is the genotype of individual 2, 4? The inheritance is showing up in every generation, and it's affecting men as equally as women. So this is going to be autosomal dominant. What is the genotype of individual 1, 1? So here's my generation. Here's the individual 1. This person is not affected, and this is an autosomal dominant trait. So I know that the genotype of this person is homozygous recessive, because if they had one capital A, they would be colored in. What is the genotype of person generation 2, number 4? This person has at least one big A because they are affected, but their father only has little alleles to pass on to this son. And so the genotype must be heterozygous. Hemophilia is a sex-linked recessive disorder found on the X chromosome. A woman and a man with normal blood clotting, hemophilia is a disorder of blood clotting, have 4 children. They have a normal son, a hemophiliac son, and 2 normal daughters. What are the probable genotypes for each member of the family? So a great practice genetic question for us, might seem really similar to one in your lab for this week. And let's draw it. All right, so we have a man with normal blood clotting. He's going to be a square. And we have a woman with normal blood clotting. She's going to be a circle. They have 4 children. They're going to be a normal son. Here's child number 1. A hemophiliac son. Here's child number 2. And 2 normal daughters. Here's daughter number 1, and here's daughter number 2. Now I'm going to draw in my lines so we can remember this is pedigree. So those 2 people had these 4 children. Now that we've created our pedigree from our information, let's figure out their genotypes. We know this is a sex-linked recessive disorder because of the text. It's on the X chromosome. So dad here, because he's a male, is going to have Y. The Y does not have any alleles on it. He's going to pass that Y onto his sons. I'm going to actually write it below because I'm going to quickly run out of space. Okay. So here's son number 1 gets a Y. Son number 2 gets a Y. The dad has normal blood clotting. So here's his X chromosome. He does not have the trait, and we know it's a recessive disorder. So it's going to be a capital B for blood clotting. We also have a woman, so she's going to get 2 X's with normal blood clotting. So she has at least 1 X of a B. Now, if we look down at her son, we know that she has an affected son. That means that the genotype of this son must be X little b. And we know that because he has hemophilia. He must have gotten this little b from mom because he got his Y from dad. That means that mom has to be heterozygous, because if she was homozygous dominant, she would not be able to have kids with Oh gosh, I can't say it. Hemophilia. Hemophilia. So now that we know their genotypes, we can assume the genotypes of everyone else. This son does not have hemophilia, so he's going to get the dominant trait. And both of these daughters do not have hemophilia, so they are going to be at least one X big B, and the other gets at least one X big B. They got both of those XBs from their dad, because that's the only thing he has to pass on to his daughters. Because they got that dominant trait, they do not have hemophilia. Now, their mother has a 50% chance of passing on the recessive allele. Since there's two of them, we can give one daughter the dominant allele and the other daughter the recessive allele, but it's the same probability each time. So there's a 50% chance for each child that they will have the dominant or the recessive allele. So either of these daughters could be homozygous dominant for the trait or heterozygous for the trait. Cool. Let's look at another practice pedigree question. Max is climbing out of my jacket. Coat color in cats is a co-dominant trait and is located on the X chromosome. Cats can be black, yellow, or calico. A calico cat is a cat with black and yellow splotches. In order to be calico, a cat must have the allele for black and yellow color. What would be the results if a female calico cat was crossed with a male black cat? Let's draw our Punnett square, our pedigree. We can do it either way. So we know that we have a female cat, so she's going to have two X chromosomes. And she's calico, so she's going to have one allele for black fur and one allele for yellow fur. That's going to give us the calico pattern. She's going to have kittens with a male black cat, like by Parker. He's going to have the allele for black fur and then a Y because he is male. What are the possible results? Well, let's put it into a Punnett square. So put a Punnett square right below that female. And then put the male over here. Cool. Now, this big B and this big B would come together and it would make a female kitten with black fur. This big B and this Y would come together and this will make a female calico kitten because it's expressing both the black and the yellow fur. This big B and this Y, that's going to be a male kitten with black fur, so it looks just like his dad. Now remember, Ys don't get the sex-linked chromosome allele. It just has nothing on it. And then the last kitten would get the yellow fur from mom and the Y allele from dad. So you have one black female kitten, one black male kitten, one yellow male kitten, and one calico female. All right, now let's look at more than one trait at the same time. The gene that controls coat color in cats is located on the X chromosome. That's what we just did. Cats can be black, yellow, or calico. The gene that determines hair length is located on chromosome 2. Hair length can be short, medium, or long. A long-haired black male cat and a short-haired feline calico cat produce kittens. Of all the kittens, what fraction of the kittens would be medium-haired black male cats? So first thing we have to do is break this down. We've got two traits. They are on different chromosomes. So we're going to either do math or dihybrid cross. Let's do the math one. We know that the cat coat color is on the X chromosome, and we have a black male cat. That male cat is going to be XB. That's his black fur, and he is male. Our female is going to be calico. So calico female. She's going to produce one X with the black allele and one X with the yellow allele. Do our Punnett square. Oops. So we have X, X, B, B. That's going to be a black female kitten. Then we're going to get a black male kitten. We're going to have a calico kitten, yellow allele on the X chromosome. This is a female. And then we're going to get a yellow male kitten with the Y chromosome being a male. So our question was, what fraction of the kittens will be medium-haired black male cats? We're answering the black male right now. How many are black male out of 4? That is 1/4 are going to be black-furred male cats. Now let's look at the second part. Long hair is on chromosome 2. We have one long-haired cat, and it's autosomal because it's chromosome 2, which means you don't need to use the X's and the Y's. You only use the X and the Y's when you're looking at X-linked, also called sex-linked. So if it's not on the X chromosome, then you don't need to use the X and the Y's. That's the biggest mistake I see of students, is they want to add the X and the Y's. So we have a long-haired cat, dominant trait, 2 of them, except we know that we have medium and short, so it's probably incomplete dominance or co-dominance. And then the female cat is short-haired, so we'll do little l, little l. Here is our Punnett square. All of our kittens are going to have one dominant and one recessive trait. The father had long hair, the mother had short hair. That means that 100% of the kittens are going to be heterozygous, which equals medium fur. So all of our kittens are going to have medium fur, but only 1/4 of them are going to be black-furred males. So that means overall, the fraction of black-furred kittens is 1/4. We could do this another way where we did a dihybrid cross. I'll show you that as well. In a dihybrid cross, you figure out the genotypes of both parents first. So we have a long-haired male cat. I'm going to use an up caret because I can't make superscript here. That's our dad. And we have a short-haired female calico. So you with me? Up caret means superscript. So pretend this is the superscript for the X chromosome. We then can figure out the gametes produced by each of these people, each of these cats. So this cat, this male, when he makes sperm, in each of his sperm, he can add the big L and then the black fur chromosome. And then he can use the first big L, but now the Y chromosome. I'm making 4 gametes of each possible version of his alleles. So I've used this first big L twice. Now we're using the second big L for long fur with his X chromosome for black coloration, and then the second big L for the Y chromosome. So it looks like it's the same thing twice, but that's because one of these is the first big L and one of these is the second big L. So those are the 4 types of gametes that can be produced by your male cat. Now the gametes that can be produced by my female cat are little L for short fur. She's got 1 allele and 1 egg, and then the next allele is going to be the allele for black fur. Then the little L for short fur and the allele for yellow fur. Now we'll use the second little L for the black fur. It's an I. And the second little L, sorry, it capitalized it the first couple times, for the yellow fur. So those are the 4 eggs that this cat could produce. Now to do a dihybrid cross, you would take those 4 gametes and you would line them up on top of a double stacked Punnett square. So you'd have 4 rows and 4 columns. And you would fill in, so this is short fur for mom, X with the big B, short fur, X with the yellow fur. These are the 4 types of gametes that your mom cat can produce. And on the other side, the 4 types of gametes that your dad cat can produce. It's a B. Long fur, that is black, and long fur with the Y chromosome. Then you drop down, you fill them in, and you count how many will give you medium furred black kittens. So all of them will give you medium fur. So you look for the black male kittens. Here's going to be your males. So these 2 columns are going to, or rows are going to be males. And then these rows would give you black fur because you're inheriting the black from your mom. You would get both traits here, 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 and here. That would be 4 out of 16, which if we reduce is 1/4 of our kittens. So 2 different ways to do it, the same outcome each time. I know that was a lot. That's why this is the lab for this week, is practicing really complex questions like this. Email me anytime with questions. Feel free to watch this as many times as you like. And I hope you have a great rest of your day. Bye.